Welcome, everyone. I'm so happy that you've joined us for our first epigenetics seminar of 2025. We're going to get started in just a minute. I'm allowing some time for people to log on. We are considering our first epigenetics in-person symposium that will be later this year. We're thinking in the fall of this year, and we'd like to gauge interest. So we're sharing a few questions with you. If you could just take a few seconds to take a look at the poll and answer the questions, we would really appreciate it. And we'll get started in just a minute. Welcome, everyone. I see people are still logging on, joining the webinar. Thank you so much for joining the first Boston Epigenetics Society webinar of 2025. We're really excited to get going. If you could take a look at our poll and take just a few seconds to consider our symposium that we would like to have later in the fall, we would really appreciate it. All right, I think I'm gonna get started. Once again, thank you to everyone who's joining us for our first 2025 Boston Epigenetics Society webinar. We have a great speaker lined up today, and I don't want to take any time from her, so I'm gonna go ahead and get started. The mission of the Epigenetics Society is to promote epigenetics and epitranscriptomics research and to foster connections between academia and industrial epigenetic scientists. I'm so thankful for our leadership steering team, who you can see here on the left. And I encourage you to reach out to us, that if you love epigenetics as much as we do, and you'd like to become more involved in promoting epigenetics, it's free. Just join us. And the way to do that is, I'll share on the next slide. I also want to thank our sponsors that we couldn't do this. Today's webinar is being hosted by Active Motif, and I really want to give thanks to Active Motif, as well as our other sponsors like Cantata Bio, Biomodal, and 10x Genomics. I want to thank them. And John Rinn will be the MC for today's event, and you'll see him there. There he is as he pops up on screen. So how can you get involved more with the Epigenetics Society? Well, we now have our own webpage, and at the bottom of the webpage, you can sign up for our email list, and you'll get all the updates from the Epigenetics Society. You can also link into us on our LinkedIn page. So we have a LinkedIn group. Feel free to email us at Boston eSociety or reach out to any of the leadership team members that I just showed on the previous slide. Today's webinar will be recorded and it'll be available on our website. So once again, just a couple of logistics. If you have any questions for the speaker, please ask them in the Q&A panel, and John will ask them of the speaker at the end of the event. If we don't get to all of the questions, Marisa may email you specifically those questions if we don't have time to get to them. Any general questions can be asked in the chat window, and we will be monitoring that throughout the talk. So without any further delay, I'd like to introduce John as the MC for today's event. Thank you so much again for joining us.
Yeah, thank you, Brian, and thanks, everybody. Today, it's a real treat to have Dr. Marisa Bartolomei with us to talk to us about her latest work on DNA methylation in the germline. And Dr. Bartolomei got her BS from the University of Maryland, her PhD at Johns Hopkins, and did her postdoc with Dr. Shirley Tillman at Princeton University, and has won tons of awards, but I'm just going to highlight a few of the big ones that I noticed. Well, Dr. Bartolomei is also the director of the Center for Women's Health and Reproductive Medicine at University of Pennsylvania, as well as the Perlman Professor in Cell and Developmental Biology. And she won the Women's Health Research Award from Medtronics, AAAS Society member, National Academy Science member, Genetic Society Medal from the UK Genetic Society, and many other international awards. But just personally, I've had the fortune of knowing Dr. Bartolomei, I think, like 20 years now. And she's just a real visionary scientist where her work is always put into a textbook and you read about it, and it takes a while before you understand how ahead of time Dr. Bartolomei's research really is. In fact, she worked on the first long non-coding RNA ever discovered, and I knew about her work before I got to meet her. And in fact, I confirmed that she still has the sequence of H19 in her office, where she found a microRNA that was later published again, like 10 years after she looked at that structure. So, and on a personal note, Marisa's just an amazing mentor, both within her lab, within her community, but even far outside of that. In fact, I was encouraged by Marisa for the first time to apply for faculty positions, which is kind of a scary thing. And Marisa always just had warmth, wisdom, support, and always made science fun. So with that, I'm just really looking forward to your talk, DNA methylation reprogramming in the mammalian germline. Thank you, Marisa.
Great. Thank you, John, and everybody for the opportunity to talk to you all. Let me see. Let's see if I get this. Everybody see this? Good?
Looks great.
Okay. Laser pointer. So again, thank you for the opportunity to give this talk about our research and to remind everybody that the Eagles are the Super Bowl champs, and tomorrow's our big parade in Philadelphia. So unless you already bought your ticket, stay away. It's going to be crazy. So anyway, let me tell you what I'm going to talk about today. The title is about mammalian germline imprinting, but reprogramming, but I just want to tell you about how we got there. Sort of our gateway to this is genomic imprinting. And I'm going to tell you a little bit about this because it gives you an idea of how, you start with this one little question and expands to other questions. Talk about germline imprint reprogramming, and then we're going to talk about TEP1, its active DNA methylation and how that works in the germline. So let's just start with genomic imprinting. And so I think probably most people know what this is, but it's mammalian specific and results in a monoallelic parent of origin specific expression pattern. So this just shows, here's the chromosome you inherit from mom, chromosomes you inherit from dad. With non-imprinted genes, you either expressed both copies from your maternal copy and your paternal copy, or repress both copies depending upon the cell type. But with imprinted genes, you really only express one parental copy. And it's always, for a given gene, it's always either the maternal or the paternal. So it's been over 40 years since the first published description of imprinting, but what we do know is really genomic imprinting is the block to uniparental development in mammals. And these were beautiful, elegant studies done by Deborah Salter, Jim McGrath, and Azim Sarani and his colleagues. And what they basically showed was that you needed to have a maternal and a paternal contribution to a zygote in order to have normal development. If the maternal was taken out and another paternal was put in, so an androgynote, unipaternal, there was a failure of embryonic development, and there were mostly extra embryonic tissues. And conversely, if the paternal copy was taken out and a second maternal was put in, so you had a gynogenode, alternatively, you can activate an egg as a parthenote, there was a failure of extra embryonic development and only embryonic tissues were detected. And so from these experiments, they concluded there was something in the maternal genome that was unique, that needed to be present for normal development, and there's something in the paternal genome that was unique. And they term these uniquely expressed genes imprinted genes. Now, that was over 40 years ago, and we know that there are a few hundred imprinted genes in the mammalian genomes. They're pretty conserved between mouse and humans, so that's been great. We can go back and forth between human imprinting disorders and using a model system like mouse to learn about mechanisms. And it just shows you some of the functions. Many imprinted genes have very important roles during fetal development. They also are incredibly important in the placenta. So there are lots of functions for these genes. And if you perturb imprinting, if you actually just takes one single hit of the actual expressed gene in order to have an abnormal phenotype. So for example, one of the genes that we work on, GRAB10, is a growth regulator, and this just shows a maternal deletion of GRAB10 results in overgrowth. And here's another CDK1C, also a maternally expressed gene that's involved in growth regulation, but here it just shows that its proper expression is important for normal placental development. And I just wanted to point out some imprinting disorders, two growth disorders, Beckwith-Wiedemann and Silva-Russell, which involve a series of imprinted genes, as well as Prader-Willi and Angelman, which are neurobehavioral disorders. And one of the really potentially exciting things about working on these disorders is that when you lose a copy of an imprinted gene, the express copy, the other chromosome has the repressed copy. And one of the ideas, and people are spending a lot of time working on, is can you activate that sort of silent copy to rescue some of these phenotypes? And it's really being done in these two diseases in particular. Okay, so what do we know about imprinted genes? Well, we know, I mean, we've known this for a long time, that they're clustered in the genome. So you can have anywhere from a few to hundreds of genes. Okay, so just briefly summarize again, these genes are clustered on chromosomes throughout the genome, and there is an imprinting control region that has differential DNA methylation. It can be methylated on either the maternal or the paternal allele, can be promoters, enhancers, whatever. And these discrete elements are critical for the imprinting of the whole region. So if you end up with if you delete this ICR, you'll see loss of imprinting of that whole cluster of genes. So the other thing that's really important about these imprinting control regions is they have these really interesting properties. So what we do know is right after fertilization, eggs and sperm, they're pretty methylated. Sperm is methylated a bit higher, DNA methylated a bit higher than the eggs. But right after fertilization, most of the genome loses this DNA demethylation. But imprinting control regions, because they're conferring parental identity, they maintain their differential DNA methylation. And what we know is that there are Crab zinc finger proteins that specifically recognize this DNA methylation, and that I think that they then help to bring in the repressive chromatin machinery that maintains that differential DNA methylation when most of the genome is becoming is being reprogrammed. And then after sort of blastocyst, you start to see reemergence of DNA methylation. Now we know that this DNA methylation patterns at ICRs are absolutely critical for appropriate function of imprinted genes. And so here's examples of these two imprinting disorders that I told you about previously, Beckwith-Wiedemann, which is an overgrowth disorder, and Silva-Russell, which is an undergrowth and failure to thrive disorder. Loss of methylation at these ICRs, or changes in methylation at the ICRs, are prevalent causes of these imprinting disorders, so dysregulation of the imprinted gene expression. So just sort of to summarize what we know about imprinted genes, we know that DNA methylation imprints are established in the germline. That's the time when these two alleles are in separate places and they can be differentially modified. These imprinting patterns are maintained during early development when there's a lot of pressure on the genome to demethylate, to reprogram, to prepare for pluripotency. And what we also know is that these DNA methylation patterns are erased in the germline in preparation for making eggs and sperm for the next generation. So this imprinting cycle goes on and on. So I told you that these imprinting control regions are very interesting in that they're some of the small part of the genome that survives this reprogramming early on. But they also and show another interesting phenotype a little bit later. So the mammalian germline is taken from the epiblast. It's designated based on its location in the embryo, and basically those cells have a sort of a somatic pattern of somatic epigenome, and because they need to be reprogrammed to become germ cell-like, there's a lot of erasure and reprogramming that goes on. And so this just shows you these cells, these germline cells, they have a high level of DNA methylation, and this methylation is then lost, and then methylation is put back on in a sexually dimorphic manner in sperm versus eggs. But what you can see is there's an early wave of demethylation and a late wave. And this early wave, it really looks like the majority of DNA demethylation, which is actually sort of a suppression of maintenance DNA methylation. So you suppress the ability to maintain it during cell division. Imprinting control regions, for the most part, as well as other parts of the genome, are demethylated in this late wave. So we know these imprinted gene control region escape these two periods of genome-wide demethylation, this point and in this point. These are primordial germ cells, the precursor to the germline, which are migrating to end up in the sexually indifferent gonad. So what has to happen for these imprinting control regions? Well, these are the precursors to the germlines. They have, this is a maternal chromosome and a paternal chromosome. Again, I'm just using an example of a maternally methylated, so the maternal chromosome, ICR. During this reprogramming, there's erasure of all the DNA methylation. And if it's a maternally methylated ICR, when these cells then enter the future ovaries, what'll happen is the DNA methylation that occurs at the maternally methylated ICRs will be put on. For these corresponding regions in sperm, we don't want them to be methylated, they're unmethylated. So that when these gametes come together, you have an unmethylated sperm and a methylated egg ICR, and that's how you get your biparental DNA methylation pattern in your appropriate imprints. So the question, as I said, how does that DNA demethylation occur? I showed you that some of it occurs by passive demethylation very early on, most is, but imprinted genes are demethylated in a slightly later phase, which is we consider to be the active DNA demethylation. I should say that probably passive keeps occurring as long as there is DNA replication, but active is really critical. So what could be the active DNA demethylation? I guess about 20 years ago, people described the TET 1011 proteins which oxidize 5-methylcytosine to hydroxy, formal, and carboxy. The 5-hydroxy can go away through passive dilution because DNMT1 doesn't recognize this as a methylated group. Alternatively, the TETs continue oxidizing, and TDG can remove the base and result in an unmethylated cytosine. TET1 and TET2 appear early on in the germline. There are 3 TETs, 1, 2, and 3, but it looks and there here you can show in immunohistochemistry of TET1 and TET2, and then we can see that when TET1 is present, 5-methylcytosine, TET2 goes away, and you see the appearance of hydroxymethyl. TET1 really does come on initially, and we've looked at TET1 and TET2, and really the star of the show is going to be TET1 for the most part. So again, when you think about the germline, only one allele has to be demethylated during gametogenesis. Again, maternally methylated ICR. And if you have a failure of erasure, this methyl group will not go away. It'll stay here. And so that when you make sperm, this is a maternally methylated ICR, which should be unmethylated in sperm. Approximately half the sperm will have that abnormal DNA methylation pattern, which possibly resulted from a failure of erasure. Okay? And so, again, we wanted to look at TET1 because it was actually expressed initially in the male germline. We and others did this. In fact, initially, we've used the mouse that the Yanish lab initially characterized to, and saw that TET1 was very important for DNA demethylation. And we look much more specifically, carefully at imprinted genes. And so these mice, these TET1 mutants, these are knockouts, loss of function mutants. You can actually generate these mice, and in fact, they are fertile. So you can then take them and study them further. But the first thing we wanted to look at, and I'm just going to talk about the male germline today. We looked at mature sperm, and what we could see, here's 4 maternally methylated ICRs. Here's 2 paternally methylated. These are methylated in the male germline, so we don't know if they're demethylated initially, but they become fully methylated, and so in a mature gamete, their methylation patterns are fine. But for these other maternally methylated ICRs, all except for SNRPN were statistically hypermethylated relative to wild type and heterozygous TET1 mutants. We also generated a double knockout for TET1 and 2, and we didn't see any difference. So we went and moved further with TET1. And what I should tell you, I just wanted to put this in really quickly, when you see these hypermethylated patterns in sperm, and we'll just focus on this one right here, KVDMR, these actually then are transmitted to offspring. So these are offspring that are generated from the TET1 paternal knockout. And what you can see is there are the average methylation pattern when you're looking at maternal and paternal alleles together should be 50%. And here we see a number of pups have hypermethylation patterns. And along with that are abnormal expression patterns of these genes that are linked. So I won't go specifically into this locus, but this just shows you if you have no TET, you will have abnormal imprinted gene reprogramming, and those abnormal DNA methylation patterns will be transmitted to offspring. Okay, so we looked at gametes, mature gametes, but the important thing is that the DNA demethylation happens weeks earlier. This is the mouse. And we never looked, we and others didn't look in between. And so the question was whether the DNA demethylation the hypermethylation we saw in the TET1 mutants, was that a function of absence of erasure of DNA methylation, or is TET there to prevent from abnormal remethylation? Okay. This has been a question that the field has had, and in part it's TET's a large protein, has lots of been shown to have a lot of binding partners, and the idea is that TET has both catalytic and non-catalytic activities, could be a scaffold for all these other proteins that are involved, that are important in reprogramming in the epigenome. And so the question is TET doing the original demethylation, or is it protecting from remethylation? So this is just sort of depicted here. So the two kinds of activities, it either facilitates demethylation, so it actually goes in and oxidizes the methylcytosine, or alternatively, something else does that, and TET comes in and protects from remethylation for regions that shouldn't be methylated. So if you knocked it out and you then did a time course, if TET was important for that original demethylation, we would expect to never see the low point in the DNA demethylation. So the knockouts would have higher levels. But if it was there for protection from remethylation, everything would go down as normal. But when the de novo DNA methyltransferases were induced after that low point of DNA methylation in the germline, then you would see increased DNA methylation in the knockouts. So we went in and we had crossed an OCT4 GFP into our lines and isolated PGCs at different stages. And I'm just going to show you 14 and a half days. It's the same for 13 and a half. And what this shows you is that indeed the knockouts never got to the lowest point. Okay, so here are XX of female PGCs, and here are the male PGCs, and you can see that the methylation level never goes down to 0 in these. So we can conclude from this that yes, TAT1 is important for DNA demethylation in PGCs. Is it important for protecting from remethylation? Probably too, but I don't have the data really to show that quite yet. So the next two questions we asked was, how does TET1 facilitate demethylation? And what I've just only talked about imprinting control regions. There's a lot of other genome. Is it involved there? So to try and address some of these questions, in collaboration with my colleague here at Penn, Rahul Kohli, we looked at generating TET1 variants that could address some of these different questions. For example, is demethylation down to 5 CAC FC required for reprogramming of ICRs and other late demethylating loci? So we generated, with their beautiful biochemistry, a 5-HMC stalling mutant that stopped at 5-HMC, didn't go through formal or carboxy. So, and the other mutant that was generated based on some of the literature and helped translating it to the mouse TET1 was a catalytic mutant. So here, in both cases, both of these proteins are full length. We initially analyzed them in a reprogramming assay, reprogramming fibroblasts to iPSCs, and we showed we needed both activities in order to have really good quality reprogramming. But what about a mouse? So we generated mice, and we did the same kind of crosses that we did before. These mice, again, are viable, so we're able to generate homozygous mutants. So, and look initially at their sperm here. And again, what we see is that for all of these, so the blue are the wild type, and then these other three are the variants. This is the stalling at HMC, and here's the catalytic mutant, and black is the knockout. So for imprinting control regions, They all look the same, okay? And we bred these mice, and similarly, we saw, again, as I showed you before, if we bred the null mice to wild type, we could see that the imprinting, the ICRs had abnormal expression patterns that were carried through to their offspring. What this is really designed to show you, here's 1, 2, 3, 4, 5 maternally methylated ICRs. For some reason, Snurpan couldn't care less if there's TET1. It uses other mechanisms to demethylate. But these 4 different imprinting control regions seem to use TET to a different degree. And so GRAB10 is a big, long, CPG-rich ICR, and it really needs TET present for reprogramming, and some of these to a lesser degree. Okay, so that, again, that's the ICRs. And the question is, there other genomic regions affected by TET1 mutations? So one of the things we wanted to look at was germline genes. And so there, you can make a really good argument why you would want to keep germline genes repressed until you absolutely need them. You don't want to sort of start producing all these meiotic proteins until you're ready to enter meiosis. And so here's an example of three germline genes. What we saw for all of the mutants, we saw they all had abnormal levels of methylation. And then when we did RNA-seq on these, we could see that this hypermethylation was associated with reduced expression of these genes. So again, that suggesting that DNA demethylation is absolutely required to activate these genes. Okay, so that was, low throughput. You can only do low throughput for so long. So we wanted to go and look at the more of the genome, and we used the Illumina mouse methylation bead chips. One of the reasons we did this is so that we could do lots of samples, because you could see that there's a lot of heterogeneity in when you looked at ICR. So we thought if we could do more samples, we could get up some better statistical significance. So we use this, excuse me, this bead chip array, and it's actually reasonable. It covers a lot of imprinted loci, promoters, enhancers, repetitive elements, trisomal elements, and a variety of other highly curated parts of the genome. And so we then looked at the methylation on these arrays. This is, again, a sperm from our 3 different mutants in wild type. And what you can see is the knockout, so the complete deletion, has many more hypermethylated regions than the catalytic mutant or the HMC stalling or HMC dominant mutant. That most of these knockout hypermethylated regions were actually rescued either by a catalytic mutant or by the stalling mutant. This just shown a little bit better here, showing the either partial rescue or complete rescue by these 2 different mutants. So the take home was that there are a lot of DNA methylation defects in sperm and the TET1 mutants, but many of them could be rescued by these various catalytic mutants. Nevertheless, there were still 556 DMRs on the array. I'm sure if you did the whole genome, you'd see a lot more that were hypermethylated in all 3 mutants. And so these regions require all that TET1 has to be able to reprogram to DNA demethylate properly. It requires catalytic and not catalytic functions, complete TET1 proficiency to making 5FC and CAC. Okay, so what are these regions? Unsurprisingly, what comes out the most, they're sort of represented on the array anyway, are these imprinted gene DMRs. So they're absolutely enriched. And you can see here that they have lots of hypermethylation. But again, it's variable. It's stochastic. Maybe if DNA demethylation starts early at a given strand of DNA, that if you have replication in place, that can help to demethylate. So you see lots of variable patterns. So what are these other regions? We call them sperm hypermethylated regions. They have imprinting control regions, as I showed you. They're evolutionary conserved transcriptional start sites. Species-specific transposons are found in these regions. So if you look at them, that here are maternally methylated ICRs in the black, and there are these sperm hypomethylated regions which really predominate. So, many more in the knockout, but there's still many in common with the other two mutants. These sperm hypomethylated regions, given that the sperm is very highly methylated in general, these sperm hypomethylation regions are very well conserved. It just shows evolutionary conservation of them. And the question is, what are these hypomethylated regions? They result from exclusion of the de novo DNMT3s due to the H3K4 di and trimethylation enrichment. It's well known that DNMT3A3L can't access the DNA to do de novo DNA methylation if the histone environment is not conducive, and HK4ME2 and 3 repels the de novo methyltransferases. And the regions that we saw that were hypermethylated in the absence of TET1 actually do have enrichment of H3K4 methylation. Now, what's interesting about this is that H3K4 trimethylation is pretty generally enriched at promoters of actively transcribing genes, but it turns out that many of our regions that we detected were not promoter associated, were not annotated promoters, okay? And we looked at this pretty carefully in the literature. We got as much, it's sort of hard to get really good, careful, purified populations of male germline cells to be able to tweak this, but in whatever we were able to show that most of our hypermethylated regions that seem like they correspond to H3K4 peaks are not not promoter associated. And this is just shows an example. This is just one of the really classic examples. So here's whole genome bisulfite sequencing at this sperm HMR, low methylation in the sperm, high methylation in oocytes. You see it has H3K4 in cotton run experiments. And in our arrays, we see wild type, no methylation. But in our 3 mutants, you see higher levels of methylation. There's 5 or 6 probes on the array that corresponds to this region. And so what we propose is maybe these are non-canonical promoters that haven't been identified because they're only used very brief time during spermatogenesis. And so we did race in adult testes for this particular region, and you can see here's the main start site, and here's an alternative start site that corresponds to this region. It's present in the wild type and not very high in the mutants. So these TET1 dependent sperm hypomethylated regions may be able to be used as alternative transcriptional start sites, perhaps at different stages of male germline development. What they're doing, we don't know because they're right in the middle of the genes. This is another example. We've done more high throughput, and it's just this. I put this in here just to show you that here's this particular gene, E158, and you can see with spermatogonian and pachytene stage, you can see different start sites using these 5' ends of the RNAs. This is just a smart STEAK2. So generally speaking, for a hypermethylated genome, the sperm genome, there seems to be very critical areas of the genome that have to maintain hypomethylation. And what I could tell you, which I really didn't include on this slide, if I take these males that are hypermethylate, that these males that are mutant for TET1 and cross them to wild type animals, we see a lot of loss we see low level of offspring, a lot of fetal demise mid-gestation. And we really feel that it's more than imprinted genes that could be causing that. So we think that this reprogramming of the genome is really important for viability, and are doing experiments now to address that. So TET1 is required for reprogramming. These are conclusions of ICRs in both male and female germline. 5-HMC dominant TET1 and a non-catalytic activity can rescue sperm DNA methylation patterns of some genes, but not at ICRs, not at meiosis genes, and not at these sperm hypomethylated regions. So it's required for other regions, and these sperm hypomethylated regions are aberrantly methylated in TET1 mutants, and these regions have HCK4 trimethylation may serve as alternative promoters. So finally, I just want to acknowledge people who mostly contributed to the work I described. There's actually been an ongoing project in the lab for many years, 2 graduate students who've since left, and then it's really being run by Rexy Prasasa, a really, really talented postdoc, and she's being helped by Jeff, who is another graduate student. So that is all, and I can stop sharing and address any questions if there are anything. I want to thank everybody for their attention.
Well, thank you. Is really you can hear like hundreds of people clapping.
Oh yeah, hundreds, thousands.
Well, there's at least 100 still here. So, and then you have some really good questions. Do you want me to start with those?
Sure.
Right. So, is DNA methylation in the germline imprinting a cause or an effect? Like, or is the DNA methylation acting as a clamp from the preceding process? So I think what they're asking is the DNA methylation sort of active, or does it follow whatever changes, chicken and egg kind of thing?
Yeah, so I mean, I think that's a really important question, right? Because when, we spend, for those of us who have been working on DNA methylation for like half of our lives, we always go around, how we see, Adrian Byrd, Harry Cedar always talk about the circle of DNA methylation and what comes first. I mean, I think that it's clear that in other processes it comes second, but I really think in the germline it may have a much more, it's possible that it has a more primary role. That said, in order to have DNA methylation, you have to have a favorable chromatin environment. So it could be a secondary to that, but I think the DNA methylation is the key for getting this sort of germline sort of reprogrammed in a great way. So
yeah, it's a hard one to know if it's passive or active. Okay, so there's lots of questions here. So so how is the gonad versus the ovary sensed for their different
oh, that's such a great question. We do not know. So it's a great question, and Rexy has some really cool ideas on how to pursue it, because I don't know if anybody here remembered Anne McLaren from she tried to do some sort of sex reversal types of things to see if you took, is it the chromosome? Is the chromosome constitution, or is it the niche? And I mean, and unfortunately she died before she could really get a great answer, but it sort of looked like a little bit of each 25 years ago before we had really good things. But I think that's a really important question. Yeah, cool question. Yep.
Yeah, I remember David Page also thought cytochrome P450s might like chop up different chemicals in the ovaries, and it's kind of complicated.
They have to know they have to know where they are. That's key. So.
Yes. Sarah Elgin is asking, very interesting, is the 5MC the only silencing mark at these sites, or do you also see marks like K9 die or try?
So yeah, so one of the things we're trying to understand is, do you have to have some sort of permissiveness for that? So I think that many of these TET1 regions are H3K9 di-tri regions. So the ones that are the most impacted by TAT1 look like they're the one. So yeah, there seems to be some overlap with that. But again, there really is an absence of really good data looking at that. You know, we don't have enough profiling of these various stages. And mostly we just need to figure out which stages we want to focus on. So yeah, great question.
And if actually to follow up on that a little bit, if canine tries mostly on centromeres and telomeres, do you see any like chromosomal abnormalities with centromere malfunction or telomere shortening?
I mean, it could be one of the reasons that we see weird, you know, a lot of atrophy. We have not looked. So yeah, is there instability? It would be a really nice thing. I'd actually, one of the things, I have Glennis Logston, who's just now my neighbor now. She's moved in to the lab next to me, and she's a centromere person. I'd love to look at mutants and centromeres with her, this kind of assay she does. We've talked about that. Yep.
Cool. All right. Is DNA methylation in embryonic development more committed and less contradictory to, say, in cancer? So maybe is DNA methylation in development more stringent, and cancer it's more plastic? Not
Probably it's probably much more important in making the germline work. I mean, I think that Tim Bester and Deborah Borkes showed really clearly that if you don't have appropriate DNA methylation and demethylation, you end up with a crisis, a germline crisis. Actually, a lot of that is because of inappropriate animation of the of a transposable element. So it's pretty important for not only for the stability of the germline, but for the reprogramming. So yeah, so I think that's the importance there. Yep.
All right.
Should be looking at the wrong question as I answered that. I saw the question above it was saying something different. So okay, hopefully I answered it.
Points to the importance of the germline development versus cancer can kind of make its way with whatever happens. This is a great question. I actually had the same question. What is the mechanism by which TET1 acts on specific ICR regions, and what is determining that specificity?
Oh, that would be so great, because everything you learn, you just move it back. You know, you move it back. So as I showed you, the ICRs, they have all different reliance on TET1. And I sort of always thought that the more CPG rich it is, the more reliant it is. So it's possible that, you know, there's some ideas of what you need to have DNA methylation, what are the right chromatin environments. You know, H3K36 transcription through the region is a signal for DNA methylation, et cetera. But I think that the question is, if you demethylate an ICR, but it still is incredibly attractive to the DNMTs to come in because it's really CPG rich or something. So it maybe there's this yin and yang. And so we really don't know where the specificity is. It's really pathetic that we don't know that yet, but we don't know it. So, and somebody asked, is it surprising that TET1 knockout is viable and fertile? I sort of alluded to that initially. So I think that what's happening is that DNA replication with a suppression of de novo methylation. So passive DNA methylation is working at the same time as the TETs. And so maybe it's compensating for that. And that's why you can get some gametes out and some viable pups. But we've looked most in the male germline, and I sort of think because there are fewer cell divisions in the female germline, that maybe it'll be more important in the female. Those mice have been harder to work with. So yeah, so it is surprising.
Yeah. And then similar to the specificity one is I'm wondering how imprinted genes are recognized to be methylated or not after erasing in PGCs. Like how do they, after it's erased, how do you know to get back there?
So the prevailing model, which it doesn't work so well, is that there are all these germline specific upstream promoters, and as transcription occurs, you have H3, you have H3K36 dimethylation that then brings in the DNMTs and activates them for methylation. So it's transcription coupled DNA methylation. And so it works. It looks like it works for some of them, but I don't think it works for all imprinted regions. And so I just, the longer I work on imprinted genes, the more I realize that it's not a one size fits all. So there's some imprinted regions where it looks really clear that you get transcription and you have DNA methylation coupled transcription, right? You get the DNA methylation put on. For other ones, it's not as clear. So we don't really understand.
Yeah, and you saw that difference with H19 in another locus that just doesn't even care.
Right, snarp bend. It doesn't care.
Yeah.
Whatever. So we don't know, and so yeah. So anyway.
Okay, so then another question is, how much is known about the mechanism for de novo DNMT3s to be excluded by HK4 ditri?
Oh, that's all been done. It
Okay.
has that histone modification has the domain on DNMT3A that basically inactivates it. So it's actually really well worked out. The same way as the HDK36 activates the DNMTs. It's sort of the two yin yang things. So that's been really worked out by other people who are smarter biochemists than me. Yeah.
All right. And then this is a good question, but kind of long one, so bear with me here. It's recently sort of appeared that gain of methyl CpG can actually lead to promoter repression, which then occurs due to abnormal K27 tri installation, especially for genes at borders between A and B compartments that are the imprinted genes in special positions of the genome.
Yeah, so there's some I have to say there's some contrasting information in the literature. Some say they are in the alleles can be found, further internal versus external. It's a mess. So it probably depends on the locus.
Yeah.
So.
Yeah. That seems to be the answer for a lot of these is they're sort of each unique or at least subclass.
Yeah.
Okay, so then what's known about TET1 binding to DNA with its activity and turnover? Or what means the protein activity and turnover of TET1?
So, so I think people are, a lot of, again, biochemists are working on it. So the question of like, is TET1 progressive? Does it, land on the DNA and just sort of oxidize? Does it stay at one residue and keep turning it over? One of the things that, people are using different mutants to address that. So that's, I think, been described biochemically as opposed to in vivo. So as long as you feel comfortable with the biochemistry, that's worked out and you can go to those papers. I'm like an in vivo biologist, so my thing is, do we need to reprogram the genome? Absolutely. So anyway. Right.
Cool. And then sort of last question. There's been lots of great talks, which I want to reiterate. And then is the DNA methylation, demethylation in germline different mechanistically and more committed to that in somatic cells?
So again, I think that's the example of where you probably see more of an interaction. In somatic cells, it may be more responding to the H3K9 trimethylation, whereas in the germline may not be quite as much yet. So again, because we don't have, we're only now starting to get sort of in vitro gametogenesis to work in mammals, and then we can start taking different times out and sort of making mutations and address that. I think that's all gonna forthcoming. So yeah.
Yeah, it seems like there's more maintenance, or sorry, more establishment than maintenance in the germline. And
Absolutely. It's much more establishment than it is the maintenance. And so, yeah. So
great. Well, I know you have to go at 11, so let's all thank Dr. Bartolomei one more time for, as always, as was mentioned, an insightful and fun talk. So
great. Thank you so much. Thanks. Nice to see everybody, and look forward to interacting in the future. Take care.
bye.